right? Polymorphism. Gauri.
Yes, sir.
So what is meaning of poly? The word poly says many, right? And what is meaning of morphism? It is morphology, right? So it's like morphology is like the outer appearance, or you can say the phenotype. There is a phenotype and genotype, right? The outer physical appearance, we call it as morphism or phenotype. Okay. So some genes, they can have multiple phenotypes, multiple outer appearance. First, suppose you just take the color of the person. There are so many colors. You can say the whitish color, the brownish color, the darkish color, the Asian color, or African color. Okay. So that means the gene itself is having, even though gene is color of the skin, but it was having so many of variations. There are multiple variations for the same gene. So we call it as polymorphism. If the gene is having only one outer phenotype, we call it as monomorphism. Understood?
Yes, sir.
Yeah.
Makes sense now.
Yeah. So what is polymorphism? It's nothing but the presence of two or more alternative forms of a particular gene. Two or more alternatives of a particular gene, like a tall plant and short plant. Okay. Like the color of the seed, or you can say the color of the plant, or the shape of the seed, wrinkled or round. So all these comes under different alternatives of the same gene. So we call it as the polymorphism. Okay. And why does this polymorphism comes? Because change in the nucleotide sequence. And this change in the nucleotide sequence, can this happen for only one single base pair, or it can happen for multiple base pairs? So based on that, we can tell it's a single polymorphism or multiple polymorphism, like single nucleotide polymorphism, SNP. So because of that change in the single nucleotide base pair, it result in causing the variation in the same species. So that result in forming the diversity. Okay. Monomorphism means it's a single form, the ancient or primitive form. But during the course of evolution, that single form was got into a different forms by the course of evolution. So we call it as the monomorphism is turned into polymorphism now. And what is the difference between the polymorphism and then the mutation? See, polymorphism will happen naturally. So this happens naturally so that the species can able to survive. Whereas the mutations, it happens due to the external factors. You can say the mutagens. So mutagens are the factors that are causing the mutations. You can say like the chemicals, or you can say the radioactive materials. So all these comes under the mutagens. So which result in change in the nucleotide sequence. And mutagens result in mutation. Mutation can give three results. One is the result will be benefit. That means it adds positive value to the species due to the mutation. So that can bring the evolution if it is that case. Second one, it can cause the destruction. So the destruction can be like mutation result in genetic disorders like the cancer, sickle cell anemia. So these are all some of the destructions. Or it can cause the neutral effect. That means it doesn't give the positive effect or neither negative effect. It's simply neutral. Mutation can result in polymorphism, but not all the mutations can cause polymorphism.
Okay. So
Okay.
it's not necessary that polymorphism is bad for the.
Yeah, polymorphism is like a diversity, like the color of the person or height of the person. Okay. So
So
that's a
what about the
diversity.
so what about people who have skin diseases?
That's a mutation.
That's mutation.
Yeah.
Okay.
Okay. So mutation, if it happens, the single nucleotide is changed and that result the positive effect, you can consider that as a polymorphism. But if the mutation is causing the destruction, then it's a genetic disorder. Diseases, genetic disorders, or diseases.
Oh, okay, sir.
Yeah. See, mutations mostly happens because of the mutagens. It can be the physical cause or the chemical cause. And that result in causing the genetic change and which brings the positive effect or negative effect. But polymorphism, something that happens by nature itself, that can cause the evolution. So there can be two mutations. One is beneficial mutations. The other one can be harmful mutations. So harmful mutations can cause genetic disorders or diseases. Okay. So whereas beneficial mutations, they can lead to adaptations and evolutions. Understood? So this can be considered as a polymorphism, but this can be considered as a genetic disorders. Okay.
Okay, sir.
Yeah. All this variation has brought the interest for the scientists to find out what is the actual genomic sequence and how does this genomic sequence was changing and that bringing the evolution or adaptations or causing the diseases. What is this 2 meters length of DNA is made up of? Adenine, guanine, cytosine, thymine. And they were arranged in a particular sequence that result in bringing the genes. So we need to find out this entire length of the sequence so that whatever, if there is some change happens in the sequence that can cause disease or that can cause evolution or that can even not cause anything, or it can also show the differentiation between the two species because of all these variations, genetic diseases, and there is a 99.9% species are similar, but that 0.1% it brings the variation in the species polymorphism. So this all brought the quest to find out SGP, human genome project. Under human genome project we need to find out the total sequence of 3 million chemical base pairs. Understood? We have two approaches. One is express sequence tag. The other one is sequence annotations. So express sequence tags. So what do we do is whatever the a length of nucleotide that is expressing. What is meaning of expressing? From the DNA we know. Okay, DNA will make mRNA and this mRNA will result in formation of proteins. So we'll take that small fragment of mRNA or the small fragment of DNA, which is expressing into the genes. And through that, you will be able to identify the total number of genes under the express sequence tag method or sequence annotation. Okay, we'll just dive into deeper. Let's see what is that. So first we'll go with the first method. So first one is express sequence tag. So we know that there is a human cell. Inside the human cell, we have a nucleus. Inside the nucleus, we have total 46 chromosomes or 23 pair of chromosomes. So in these chromosomes, the total 2 meters length of DNA is aligned. Yes or no? So which is having almost 2% of genetic information and 98% of non-genetic information. That means 2% only DNA will make the proteins. The entire 98% of the DNA, it's just not making any genes, not making any proteins. It's like you can say junk DNA. So we need to find out all this. Further, first we need to isolate the human DNA from the any particular cell. So once we isolate it, we know that this DNA, it contains both the coding part and non-coding part. The coding part, we call it as the exons. The non-coding part, we call it as the introns. Yes or no?
Yes, sir.
Yeah. So we need to sequence all this coding part and non-coding part. Okay. So coding genes means that which will be expressed as mRNA. Yes or no? So DNA will make hnRNA. That hnRNA will contain both the coding part and non-coding part. So in that, if we remove the non-coding part, we left with the coding part. That coding part is nothing but mRNA, right? That means whatever the junk DNA is there, you remove that, it will result in making the mRNA. So that mRNA is the one which is expressed part. That means it can make the proteins. But if you take the hnRNA, it contains both the expressed parts as well as non-expressed parts. That means the coding part as well as non-coding part. So during this approach or during this method, you will be removing all the introns. You will be eliminating all the introns and you are considering only the exons or the mRNA. So remember, during the express sequence tags, all the introns were removed. We will not sequence the introns. We will only sequence the exons. Now tell me, so Will I get the total entire DNA, or will I just only know the gene part of the DNA during by this method? Will I understand all the DNA sequence, or will I only able to understand what is the gene part?
Only the gene.
Gene part.
Only the gene part. Remember this, we are removing all the introns. That means you are removing the junk DNA. That means 98% of DNA, you are just removing it. Only 2%, whatever that is coding for the genes, you are considering it for this method. So that means only the gene part, which is nothing but the exons, which is 2% of the DNA, total DNA. So remember, express sequence tag will not give you the complete picture of the DNA. It will give only the picture of whatever that is coding. Whatever the gene part is there, it will only tell about that part, not the 98% of junk DNA. So remember, we will be removing the introns. So left with the exons itself. So hence exons are sequenced and analyzed. Whatever the introns are there, the non-coding part, they were removed. And we will not study this part in the express sequence tag method. So express sequence tag method only studies about the gene part or whatever the coding part is there. That can be sequenced and analyzed. It will not study the non-coding part. So remember this method. So see, so only expressed genes are sequenced, therefore exons are sequenced and analyzed. So this process doesn't give the complete genomic picture, but only gives the expressed regions or the gene regions. Okay. So how do you do this method? Just a second. Okay, in this PPT, I didn't give you how we'll do express sequence tag. I just removed it, but I'll just in rough, I'll tell you. So what is express sequence tag? What do you understand by this?
It's the gene part of the DNA by
Okay,
removing the
so
intron.
yeah, the first we need to take any cell. Okay, so take any cell. So from that, we need to isolate the DNA. Okay, so actually what we do is, so every cell, let's take any cell. Does this cell will create all the genes in a single go? Will it make all the proteins, whatever? I told you that there is 30,000 genes were there. In that, we know 15,000 genes, what is their function. And whatever the 15,000 other genes are there, we don't know what is their function. That means whatever the 15,000 proteins that our body is making, we don't know actual function. We just only know the 15,000 genes, what the protein use is. But we don't know the other proteins. So now tell me, does any cell at a given point in time, can it make all the 30,000 proteins in a single go?
No, sir.
No, it will not make. It will just only make few genes. Let's say some 5 to 10 genes it is making. Okay. So that 5 to 10 genes, for that, whatever the DNA is there, it will make the mRNAs. So they will make 5 to 10 different mRNAs. From those 5 to 10 different mRNAs, we'll be getting 5 to 10 different proteins. Let's suppose at just now, right now, I'm measuring my beta cells of Langerhans. So it is making 5 different proteins now for performance of this cell. So these 5 to synthesize 5 proteins, first it has to make 5 mRNAs, and those 5 mRNAs has to translate so that you can synthesize 5 proteins. Yes or no?
Yes, sir.
So if I take this complementary mRNA, I can understand what is the nucleotides is made up of for these 5 proteins. I'll get a snapshot of the genes that can synthesize these 5 proteins. Yes or no?
Yes, sir.
Yeah. So we know mRNA is nothing but it is made from the DNA, right? So DNA will make hnRNA. From hrNA, if I remove all the introns, I can make the mRNA. That means it gives, and this mRNA will give the protein. So this mRNA will just give me the snapshot of this protein. I mean, in the sense of what is the nucleotide sequence for this protein. Yes or no? So to synthesize this protein, at least I'll take some 10,000 or 20,000 or on an average 30,000 nucleotides. So for this 30,000 nucleotides, I can synthesize this protein. So I can understand what is the sequence for this protein by understanding the mRNA. Yes or no?
Yes, sir.
Yes, sir.
Yeah. So what I do now, I'll take this mRNA. From that mRNA, I'll put that into a bacteria cell. Okay, and I'll multiply that, and I will understand what is this mRNA is made up of. The same techniques, whatever we did in DNA fingerprinting, we'll do that one. So for that, we'll study in detail in the next chapters, the biotechnology chapters. Right now, I'm not going into deep. Okay, so just understand this one. So whatever the mRNA is having the nucleotide sequence for synthesizing a particular protein, we are amplifying that mRNA and we are studying that mRNA, and we will get a nucleotide sequence to synthesize this protein. So like that, you will do for every each and every protein. For suppose beta cells, I will be able to understand the sequence of the insulin. I'll take thyroid and I'll find out the sequence for the thyroid hormone. I'll take epinephrine, norepinephrine, estrogen, testosterone. So like that, every protein I will identify by using this technique. So like that, you will be studying each and every gene, and you will be having the nucleotide sequence for all these genes by this express sequence tag method. Okay. So this is only specific approach. That specificity will be only the gene part or the coding part. Whatever the non-coding part is there, you cannot study this one. On the contrary, the sequence annotation will be giving the complete blind picture of the entire gene. The total 2 meters length of gene it will give. Is that clear?
Yes, sir.
It's okay. You don't need to understand each and every step, but just understand that only this point. In the express sequence, we will only study and analyze the exons. We'll not study the introns. That means we will get information only the gene part. Whatever the non-gene part is there, the express sequence tags doesn't give the information. That much you need to know. Whatever the extra information, I'm just telling you so that you can remember it. Okay, only this point is very important in the express sequence tag. Next one, we have the sequence annotation method. So sequence and annotation. First, there what we did, first we identified the protein, and this protein is made up of particular mRNA, and this mRNA is having particular nucleotide sequence. That's how we discovered. So this mRNA will be having particular sequence of DNA. So like that, we have did for each and every protein. So that's the previous method. But in this method, what we'll do is we'll take the sequence of the DNA. From that sequence, we'll understand what is this making mRNA. I mean, this DNA is making mRNA. From that mRNA, what are the proteins you are getting? So we will go this order for the sequence annotation. DNA to mRNA, mRNA to proteins. But in previous method, first we'll see what is the protein, and this protein, what mRNA does it have? From this, what is the DNA sequence does it giving? That's the previous method. But here, first you sequence the DNA and then you mark it by what protein it is giving. First, suppose I have total 46 chromosomes. On my 23rd chromosome, at the particular region, it is giving this gene. So that's how we are doing in this method. In the previous method, what we did, I just extracted, this is the gene I'm getting. From the gene, this is the mRNA we have. This mRNA is having this DNA. So that's how we did. Now what we are doing? So we are taking any particular point, and from that point, what is the mRNA it's giving? And from that place, what is the protein it is giving? So first you need to sequence and then you need to mark it or annotate it. So let's see how does this will happen. So the same thing, first you will take the DNA and you will chop down the DNA into small fragments. Okay. So take the entire length of DNA and cut down that DNA into small fragments. And whatever the small fragment is there, you put it in the bacteria or you can put it in the yeast. So do that. Okay. So we call this method as a cloning method. That means why we are inserting these small fragments into either a bacteria or yeast to make it into a multiple copies. So why we are doing this? To make multiple copies. So remember, the first technique is isolate the DNA, and the second technique is cut down the DNA into small fragments. And third step is put that small fragments into each and every bacteria and make it a multiple copies. Is that clear?
Yes, sir.
So why
Yes.
are we making multiple copies?
I'll tell you what is the purpose behind that. So for that, I said we can put that in the bacteria or you can put that in the yeast. Okay. So why? To make the multiple copies. For that, we are using BAC or YAC. That means bacteria artificial chromosome vector or yeast artificial chromosome vector. And what is vector? It's nothing but the vehicle. So the vehicle that carries our small fragment of cut DNA. I chopped the entire length of DNA into small pieces, right? And this small piece I inserted into the bacteria or yeast. For that, I need a carrier that is the vector. That we'll study in depth in the biotechnology chapter, how we are transforming, how we are creating the modifying organisms by using biotechnical methods. Okay. So right now, just remember vector means it's a vehicle or a foreign material, foreign DNA that one can carry. So remember that one. So you can use that as a BAC or YAC. And what is the mechanism? If you ask, the mechanism is simply like, do you remember the bacteriophage? The experiment we studied, bacteriophage. So we are inserting the virus into bacteria phase, and the bacteria phase can form radioactive protein or radioactive DNA.
Yes, sir.
Okay, the same mechanism will be happening here. It's okay. That will be studying in detail in the biotechnology chapter. Right now, just remember this one. Okay, the same mechanism we are using here. Okay, so BAC is a bacteria artificial chromosome. What is this artificial chromosome? All that you will study there only. So you just keep it at time. Okay, just understand this. So how do we do is we take this a vehicle, that's a vector, and this is the small fragmented DNA. I told you now, whatever the total DNA is there, we are chopping into small pieces. And that one of the small piece is this one. So the DNA fragment. And this is the entire length of the vector DNA. So what I do, I need to insert this small fragment DNA into this vector. So for that, I need to cut down this BAC. So whatever the circle is there, now it was unwinded by cutting down here. So once I cut down, so with the help of DNA ligase, you know, right? Ligase enzyme will act like a glue. It acts like a gum. So this will attach the DNA fragment to BSC. So you can see this picture. This is the foreign DNA that we inserted into the bacterial vector or artificial chromosome. Okay. And why does we do this method? For amplification. Where do we studied amplification method? I think I think I didn't teach you anywhere. That I'll teach you in the biotechnology chapter. Right now, just remember. So there will be bacteria. Yeah, at least you remember this one. There is a bacteria. So how does the virus will reproduce? So virus will send its DNA into the bacteria or host cell. And this host cell is having its own genes or own DNA. So this viral DNA will mix up with the bacterial DNA. So whenever the bacteria is making the proteins, you will also get the viral proteins. Yes or no? That's how the bacteriophage acts like a host. that we studied in the experiment also.
So, sir, this is similar to lytic cycle, right? Of
Hmm.
viral replication.
Yeah. The same thing happened. The mechanism is same, but instead of viral DNA, we are inserting our gene of interest. That means
Okay.
our fragmented DNA. That's it.
So we are following the same cycle of viral
Yeah,
replication cycle.
yeah. So you will be amplifying that one. So this DNA, whatever is there, it will replicate and form many number of copies. The same way, whatever the small fragment we are inserted, that can also will form multiple copies. It's okay if you don't understand this very well, no problem. For this, you need much more foundation or basics that we'll be studying in the biotechnology chapter. What is amplification? How do you use gene of interest? That's a complete whole chapter is there. So right now you just understand. So we are amplifying. For amplification, we are taking a bacteria or BSC and we are inserting our gene of interest or our fragment DNA into this so that this can make a multiple copies. That much you understand. Mandara, you got it?
Yes, sir.
Okay. So after this step, you will get a lot of fragments of the same gene. So we call it as amplified DNA fragment. Initially, whatever the DNA which I took from a single cell, I chopped down into small pieces, and one of the piece I inserted into BSC. Now this BSC has made this small fragment into many copies. That's what happened. Okay, amplified DNA fragment. That means the same copy we made into multiple copies. We duplicated it. We cloned it. That's why it is called as the clone. That means whatever the same one is there, you are making a multiple copies. So we call it as a clone. So see here, along with the DNA fragment will replicate. That means the BSC, the host bacteria will replicate. With that, whatever the fragment we are inserted, that will also replicate for several copies. And whatever these several copies that are replicated, we will isolate them and we will sequence them. That means we put them in an order. And how do we put them in an order? We use that by bioinformatics. We use the specialized computer program and we'll order them. So see here, this is the small fragment which I cutted and inserted. So from that, I made a several copies now. I amplified it. You can see this one. So we cannot do that. That can be done by the computers. Okay. So computer will sequence it and will arrange it. So once it will arrange it, you can able to synthesize HNRNA then. See, we are going now. Okay. So that's a exons, introns you will be getting. So once the genome is sequenced, the different regions of the genome are annotated or marked. That means which is the coding part, which is the non-coding part, you will be mark them. Okay. So once you do that, so you will assign which uh, particular region does this DNA belongs to. So you will assign that into a chromosome. So that's how you will do for the entire chromosome sequence. Not only that, in that sequence, where does this small fragment is present, you will also do that. Okay, that we call it as gene mapping. So that means the process of assigning the position of the genes in a particular chromosome is called as gene mapping. Okay, first suppose on my second chromosome, at this region, my hair color is present. We have two set of chromosomes, right? One is from the paternal chromosome and one is from maternal chromosome. The same region, my mother chromosome will also contain the hair color. At the same region, my father is also containing the hair color. And based on which one is dominant, which one is recessive, I can get that color. If my father's is dominant, I'll get my father's color, like black one. If my mother's is dominant and my mother is a somewhat reddish color, I'll get the reddish hair. If both are dominant, showing the co-dominancy that we'll be studying in the principles of inheritance, then I'll get the mixture of black and red. It is neither black, it's neither red. I'll be getting the intermediate one. Okay. So that's how gene mapping will be done. So you can see here, there are different gene mappings for different genes. So this is one gene, this is one gene, this is one gene. Okay. So position of different genes can be found out on the chromosomes. And whatever the remaining part is there, that's completely non-genomic part, or you can say the introns part. And remember, so by this project, we have understood that there are total each gene on an average contain 3000 bases. I mean nucleotide bases. So the biggest human genome is dystrophin, which is containing 2.4 million bases. And this dystrophin protein is helpful for recovering the whatever the damaged muscles are there. Okay, so whatever the damaged muscles are there, they have to recover. For suppose let's say Mandara is doing TikTok and her leg is slipped and she was fell down. Her muscle fibers were damaged at her ankle, so it has to recover. So for that, we need a dystrophin protein. If dystrophin protein is abundantly present, she will recover soon. If it is not present, she will not recover at all. So when the people getting old, the recovery will be getting decreased. So strengthening of the muscle fiber and protecting from the injury, we need dystrophin protein. So that's one of the largest human gene, which is having 2.4 million bases. So like that, with the help of this HGP project, we have discovered total 30,000 of genes are present, but in that only 50% of genes were known, and whatever the 50% of genes, they are yet to be discovered. That means they are synthesizing the protein, but we don't know what is this protein function is. That means we only know 15,000 proteins function, and whatever the other 15,000 are there, they are yet to be discovered. And if you see this total length, 30,000 genes, they only make less than 2% of the entire genome. That means whatever the 98% is there, this 98% is not at all coding anything. Only this 2% is just coding, and in that you are getting total 30,000 genes. In that only 15,000 genes were known, 15,000 genes yet to be know what is their function. And if you see this 98% genes, they are mostly repetitive sequence, like repetitives of several thousands, hundreds of A, T, 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 G, 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 C, C, C. And some are
Sir,
having the repetitives for several times, like say AGC is having several repetitives. So we know that's are called as variable tandem repeats, which will be helpful for identifying the
sir,
species. Yes, 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 you are asking something.
yeah, sir, since only 15,000 were discovered
Yeah,
and 15,000 were undiscovered,
yeah.
does that mean human genome project is still pending? Like it's not complete.
Yeah, it's not completed. We need to discover it.
We still need to discover. So you
Yeah.
can't say human genome project was like 100%
I
successful.
mean, yeah, they did from this age to this point and they discovered. I think even this was ended by 2004 or 5, right? So by this time,
It
20
ended in
years, they might have
2006.
discovered few more. It's like a going project. It's not the ended project. Even we don't know completely about the total DNA. No
Oh,
scientific study is just ended. It will be like continuing. So if you dig, you will find much more.
okay.
So whatever that so far you studied, it can be even wrong also, until the new thing will come and prove it.
Oh,
That's
okay,
how
sir.
everyone believed that sun is revolving around the earth. But later on, when he said earth is revolving around the sun, everyone laughed at him. I don't know what is that scientific scientist name, but later on
Galileo.
that was the truth.
Galileo.
Yeah, Galileo. Yeah, yeah. So even whatever that we are studying, still we don't know. I mean, this is the mostly understood one because it has the scientific reasons also. But still, at some point there can be mistakes also, which will be, uh, I mean, which will be found in future.
So human genome project has not properly ended. It's still going.
Yeah. Why you want to get into this project?
No, sir, I was just asking.
Yeah. Okay.
Because during the start, you said that it started in 1997 and then it ended in 2003, and not 1997, somewhere before that. But
Yeah. Yeah.
that I got confused. That's
Okay.
why.
Okay. No, no, no. It's like they did this 1 decade of study and they discovered all these things.
Oh,
Yeah. They
during that
stopped it.
decade.
Yeah. For suppose, let's say, like they group of people came together and they started, and at the end of the day, they by this project they discovered it. But someone can still continue that project. But mostly
Oh,
by the
okay.
completion of this time period, they all discovered that and they ended there because people can't study 100 years. They want to retire and they want to live with their family. I'm just
Oh,
kidding.
okay, sir.
I'm just kidding. That's not the true reason. You might think.
Okay.
And yeah, my friend is doing this bioinformatics, MS in USA. She is doing this.
Oh, okay.
Yeah. It has a very good career in the US, bioinformatics. Next one. Mhm. Yeah. So whatever the repeated sequence, they may stretch to sometimes several of thousands, hundreds times. Okay. And remember, in that we have a adenine and thymine which were bonded with a double bond, and guanine and cytosine they are bonded with triple bond. And if my some thousands of sequence which are just only A and T, okay, adenine thymine, adenine thymine, adenine thymine, then what happened? All this region we are just only having double bond, and double bond is a weak bond compared to triple bond. So this is the region where you can form the replication fork or replication bubble, because it is having the double bond and double bond is a weak bond. So it is a delicate region where you can split the DNA. So AT rich region will have less strength because of only two hydrogen bonds. So compared to GC. So function of this region is replication bubble. We have studied during the DNA replication, right? So there is a formation of replication fork by breaking down the hydrogen bonds. And this can happen morely in this region. So most long DNA repeated sequence have no function, but they will just only provide the structure to the chromosome. So whatever the 98% genes are there, I mean 98% of DNA, it just act like a bulk. It creates the bulk for the DNA, or it acts like a structure for the chromosome formation. So in that structure, it whatever the genes are there, they were dispersed entire 46 chromosomes. So why to have this one? First, suppose if I just put the 2% concentrated DNA in my nucleus, what happened? If something happened, there is a more frequency for this 2% to easily get affected. But if I put 98% non-genetic part, and in that, if I arrange my 2% of DNA around all the DNA, so when something affected, we can conserve this 2% without affecting anything. So that's the reason why 98% is just a junk part and 2% is the coding part. To protect this 2%, we are covered with the entire junk along with it. So by this human genome project, we got the applications like we can use easily identify the diseases, genetic diseases like sickle cell anemia. That means we know most of the people will be having CTC, that codes for CTC GAG will code for normal red blood cell. But the person where T is mutated and formed into A, we call it as defective red blood cell. And remember, this is like a polymorphism, but polymorphism will always create benefit to the nature. But this is not creating benefit to the nature. So we will not take it as a polymorphism. We'll call it as a mutation. Okay. If there is change in this single nucleotide base pair and that result in causing the beneficial effect, we can call it as polymorphism. Anyways, polymorphism means it is making unusual other pair. That's it. Or unusual other phenotype. You can say this one as well. So the person who is having normal red blood cell and the person who is having defective red blood cell. So the same gene, but two different kinds of forms. One is the good form, the other one is bad form. So we can call it as polymorphism, even here. So with this help of genomic sequence, whatever the analysis of changed base pair, we can easily identify and we can try to protect that one. So advanced treatments can come and can change these nucleotide base pairs. And you can even identify the cancer cells as well. So we can identify the genetic sequence for the cancer growth in the normal cells. And we can neutralize that effect. And we can also do that for forensic. And these all will be studying we can make a different hybrid forms from compared to the original form and make more productive by increasing the quality of the crop, quantity of the crop, pest resistance. Even it can it can grow in the lack of water also. You can make the crop very strong by using these projects. That we'll be studying in the next chapter called biotechnology. The entire chapter is there. Two chapters are there: biotechnology process and principles and applications. So we'll be studying in detail how can we do all these things. So that's it. We ended up.
Sir, when will we learn about